Imagine a microscopic world where life finds a way, even in the most sterile environments imaginable. This is the reality unveiled by the resilient bacterium Tersicoccus phoenicis, a tiny organism that's causing big waves in the scientific community. It's challenging everything we thought we knew about sterilization, particularly in high-security environments like spacecraft assembly cleanrooms. These facilities are designed to be virtually germ-free, as even a single microbe could hitch a ride into space, potentially contaminating other planets. But here's where it gets controversial. Tersicoccus phoenicis has proven that surviving these conditions is not just possible, but perhaps more common than we realize. What exactly is Tersicoccus phoenicis, and how does it pull off this incredible feat? This bacterium, belonging to the actinobacteria group, was first discovered in clean rooms at NASA's Kennedy Space Center and a European launch facility. The key to its survival? A dormant state, also known as a viable-but-not-cultivable (VBNC) state. This means the bacteria remain alive but don't grow under standard lab conditions. Think of it as a biological hibernation. The study published in ASM journals revealed that nutrient deprivation and prolonged drying, common in spacecraft assembly areas, trigger this dormancy. The team found that the lack of spore formation doesn't prevent its persistence, showing that this dormant state is more flexible than previously believed for non-spore-forming microbes. So, how does this bacterium remain hidden? Initially, scientists noticed a drop in colony-forming units, leading them to believe the sterilization procedures were effective. However, microscopic analysis revealed a stable total cell count, indicating that the cells were simply not dividing. The researchers then revived these dormant cells by adding a resuscitation-promoting factor, a protein known to awaken related bacteria. Once exposed to this factor, the previously undetectable cells resumed growth, confirming they hadn't died. They were merely dormant. This highlights a critical gap in detection methods that rely solely on immediate growth, potentially allowing other environmental microorganisms to escape detection during routine sterilization checks. Why does this discovery matter for planetary protection and clean room safety? Space agencies have strict planetary protection policies to prevent contamination of other planets. The ability of Tersicoccus phoenicis to stay dormant under these conditions challenges the assumption that only spore-forming bacteria pose contamination risks. If such organisms survive assembly procedures and remain viable during interplanetary travel, they could revive once exposed to more favorable conditions on another celestial body. This poses a threat to scientific integrity by creating the possibility of false signals in life detection experiments. The implications extend far beyond outer space. Many industries on Earth depend on reliable sterilization protocols, including pharmaceutical production, hospital operating rooms, and food processing plants. If bacteria like Tersicoccus phoenicis persist undetected in dormant states, these environments may not be as sterile as testing suggests. This discovery is reshaping our understanding of microbial persistence. Traditionally, dormancy has been associated with spore-forming bacteria that can withstand extreme conditions. The recent findings demonstrate that other bacteria can also adjust their metabolic activity to survive environmental stress without forming protective spores. The study encourages a renewed focus on molecular methods that detect DNA or RNA directly, rather than relying solely on growth-based tests. It also introduces the possibility of using resuscitation-promoting factors to reveal the presence of viable dormant cells. This dual approach may substantially improve the accuracy of contamination assessments in environments where even a single surviving organism is significant. What does this mean for the future of clean room standards and biological monitoring? The resilience of Tersicoccus phoenicis suggests that spacecraft assembly facilities may need more advanced microbial monitoring systems. Protocols that combine molecular sequencing, metabolic markers, and resuscitation-based assays could identify cells that appear absent in traditional testing. As planetary missions become more ambitious and target locations such as Mars, Europa, or Enceladus, the prevention of forward contamination will require increasingly sensitive detection strategies. In terrestrial settings, industries that depend on precision sterilization may also benefit from reassessing their verification methods. Dormant bacteria could persist on surfaces that pass routine cleanliness tests, and improvements in detection could help prevent contamination events that impact product safety patient outcomes, or research quality. The discovery reinforces the idea that microbial survival is often more complex than expected, and that successful sterilization involves more than eliminating cells that are immediately cultivable. As scientists continue to explore the behavior of Tersicoccus phoenicis, the insights gained are likely to influence how sterility is approached across both space exploration and high-security cleanroom environments. What are your thoughts? Do you think current sterilization methods are sufficient, or do we need to rethink our approach? Could this discovery change how we approach sterilization in everyday life? Share your opinions in the comments below.